talk about the types of biomolecules. The first one we're going to talk about are lipids. Compounds, they can include fats, oils, and waxes. They are insoluble in water. And they are responsible for fingerprints that are left behind, which is a very important tool in forensic chemistry. Waxes are a type of lipid, and they are a hard solid at room temperature, and they are formed by a fatty acid and a long chain alcohol. So here is an example of a fatty acid right here. The carboxylic acid portion is here, and then the CH groups are here. This is kind of known as the fat part of it. This is known as the acid part of it. So it is a fatty acid, and then it reacts with this long chain alcohol to form an ester right here. And you can see that there are a lot of CH groups in here. Triglycerides are another type of lipid. And this is glycerol reacting with three fatty acids. So here is glycerol right here, and it reacts with three fatty acids to make this triglyceride. So triglyceride is also a type of lipid, and we have two types of triglycerides. We have fats, which are semi-solid at room temperature, so something like butter or Crisco, though, or just something like butter is a fat. It is a semi-solid at room temperature. A fat is going to be a saturated fatty acid. And a saturated fatty acid means that there are the maximum number of hydrogens bonded to each carbon. In other words, there's only C-C single bonds. There are no double bonds. So that is going to be the definition of a fat. An oil is going to be a liquid. It is another type of lipid. It's a type of triglyceride. An oil is an unsaturated fatty acid. Not have the maximum number of hydrogens bonded to each carbon. In other words, you have the presence of C-C double bonds. When you see this kink, that is evidence of a double bond. Here we have two double bonds and we see two kinks. So the more double bonds you have, the lower the boiling point is going to be, or the lower. So it's going to be more like a oil and take some of the hydrogens off of it. Sorry, you can take an oil and you can add hydrogens to it to get it to be saturated. And that's how you can get some of these oils that are solids at room temperature. And in doing so, you can get a solid fat that is not from an animal. Generally, fats are going to be sourced from animals, whereas oils are generally going to be sourced from plants. So things like margarine or butter substitutes are going to be oils that have been partially hydrogenated, which means that you've added hydrogens to some of these double bonds to make it not to make it more saturated. So again, the more double bonds you have, the more likely it is that you're going to have a liquid rather than a solid. Another type of biomolecule is a carbohydrate. Carbomolecules, and they contain only carbon, hydrogen, and oxygen, and they have the general ratio of one carbon, two hydrogens, and one oxygen. When you have a molecule that has a bunch of these OH groups on almost every single carbon within this ring, then that is called a hydrate. So a carbohydrate is where you have all of these OH groups, OH group, but the ratio is one carbon, two hydrogens, one oxygen. These are also known as saccharides. They're also known as saccharides. Now, there's all different types of saccharides. The most simple is a monosaccharide. This is sometimes known as a simple sugar, and there is only one sugar unit in a mono. A disaccharide is two monosaccharides that are going to be bonded together. So another way of saying that is we have two sugar units, if you will. Here are some examples of sugar units. We have glucose, galactose, fructose, ribose, and deoxyribose. Notice the difference between ribose and deoxyribose is ribose has an OH group right here, whereas deoxyribose does not have an OH group. You just have the hydrogen. So deoxy kind of stands for you've taken away an oxygen from ribose. A sugar is going to refer to any monosaccharide or when we think of sugar in our everyday life, we generally think about the sugar that we use for baking or that is found in sweets, but in biochemistry, it's going to be any sugar molecule. Some other types of saccharides we have are oligosaccharides. And this is where we have 3 to 10 monosaccharides. And we can also have a polysaccharide. To even thousands of individual sugar units. Now, these individual sugar units are called sugar monomers. And when they all bond together, we call that a polymer. So, a type of polymer is going to be sugar monomers. So, a polymer is when you have multiple units bonded together, and those units are generally going to be monomers. For saccharides, for polysaccharides, they're going to be constructed of the sugar monomers. Cellulose is an example of a polysaccharide, and it is made up of glucose, individual glucose molecules. Cellulose is found in plant cell walls and in cotton, and humans cannot digest. Starch is also made of glucose. However, it is bonded together in a different way. And what's so amazing about this is it's also made of glucose. We can't digest cellulose, but we can digest. And starches are generally a dietary source of energy. They can be found in potatoes and rice. Another type of biomolecule are amino acids. And amino acids are going to be made up of two functional groups on a central carbon. That central carbon is going to be bonded to an amino group, H2, and then it's going to be bonded to a carboxyl group, COO. And so it's an amino acid because this is a carboxyl group. It is a carboxylic acid. And then we have the amine group. So it's amino acid when you put them together. If you have two amino acids or more that are bonded together, we call this a peptide. So if you're dealing with a peptide, it means that you have two or more amino acids that are bonded together. Amino, we have several amino acids, and they are defined by what is called the side chain. This example that I've given up here is the most simple amino acid. It is glycine. Here we have a table of different amino acids, and you'll notice it's the same structure. The only thing that changes is this R group, and it's going to be referred to as the side chain. And when the side chain is a methyl group, so CH3, then we have alanine. You can see that there are all different types of amino acids, and it's all going to depend on these side chains. When you have 10 to 20 amino acid monomers bonded together, this is an oligopeptide. It is an oligopeptide. Oh man. If you have between 20 and 50 amino acids, this is called a polypeptide. 20 to 50 amino acids is going to be a lot of names, and they're going to have a very specific order. And that order is going to be dictated by listing the amino acids in the order that they are bonded together. Since we don't want to just write out the name of every single amino acid, we have these three-letter abbreviations we can use for the amino acids rather than writing out each name. So if we wanted to do an example of leucine bonded to lysine bonded to glycine, instead of writing out each one of those amino acids, we can use the three-letter abbreviation. So leucine is LEW. Notice the first letter is capitalized, and then the next two letters are lowercase. You put a dash in between the different amino acids, and then we would write LYS. Notice again, my first letter is capitalized and the follow two are lowercase. And then finally, if we wanted to do glycine, we would do GLY.